Hi, my name is Shemi. I work for the Genomics Platform, which is part of the Broad Institute. I graduated from Boston University and I have a degree in biochemistry and molecular biology. I am a process development associate and I specialize in library construction. And basically what we do is that we process the samples sent to us through a series of steps, starting from plating. Occasionally we'll do extractions, however, that it's a specific type of product, which we do have. From plating, we get it to a specific concentration, in which will be then taken to library construction. And what happens there is that we first site select for different fragment sizes, depending on whatever the product is. And then from there, we basically clean up the DNA and do a series of formulas which optimize the DNA strands and then amplify. From amplification, at this point, the samples are good to go. And from there, what you would do is do quality control. So we do a great deal of quality control. First, we quantify each sample coming out of library construction to make sure that they have passed our standards. That would then ensure that our hybrid selection goes well. From then, before we hand it off to hybrid selection, we go ahead and we plex or pull all these samples together. And we're able to do that because each sample has a different barcode, as basically which we gave it to during library construction. At this point, what we would do is that after plexing, we take a small aliquot. We then normalize it down to a concentration that is acceptable for a smaller sequencer called MiSeq, and we do that in order to see how even our plexing was. From there, what we would then do is check how even our pull was, and if it wasn't very even, we will go ahead and pull correct by adding more of the original sample onto the actual pool, and from then we go ahead and we hand it to hybrid selection. After it goes through hybrid selection, it goes through another quality control in which we make sure that, or at least try to make sure that everything went well, and we again quantify everything, and then we pass it on to sequencing, and basically before then it's denatured in order for it to align with the DNA sequence, with the sequencer, and from there we'll eventually get data that would be delivered to you or the collaborator.